Hello, knowledge seekers. In this episode of 20-Minute Books, we dive into The Epigenetics Revolution by Nessa Carey, a remarkable exploration of how our genes are not the sole architects of our destiny. With her background in virology and expertise honed as a former senior lecturer in molecular biology at Imperial College London, Nessa Carey illumines the intricate world of epigenetics, where factors beyond our DNA sequence influence gene activity and expression. Discover the profound insights into how genes and environment intertwine, shaping everything from mental health to obesity, an unveiling of biology's dynamic interplay with our daily lives. As Carey expertly guides us through this scientific frontier, The Epigenetics Revolution is an essential read for anyone captivated by the cutting edge of biological science, from ardent science aficionados to those seeking conversation sparklers with the power to impress. Join us to unpack how the unseen mechanisms of epigenetics may play pivotal roles in understanding human development and disease. Whether you are a sage of the dinner party circuit or an amateur psychologist curious about the impact of trauma, this book promises to expand your horizons on the influence of epigenetics in our lives. With Nessa Carey's other literary contributions like Junk DNA and Hacking the Code of Life, her expertise is sure to engross and educate, proving yet again that the story of our genes is far more complex and compelling than we ever imagined. The Epigenetics Revolution: How Modern Biology is Rewriting Our Understanding of Genetics, Disease, and Inheritance. Introduction: The genetic treasure map that's more than meets the eye. Imagine it's the turn of the millennium, and the air is buzzing with a collective sense of a scientific breakthrough that rivals landing on the moon. Scientists have mapped the human genome, the genetic recipe for a human being. This colossal charting reveals not just the threads of our biological tapestry, but promises we might now begin to understand the patterns they weave. But as the fanfare fades, a question lingers. Is knowing our genes the same as knowing ourselves? Enter the realm of epigenetics, where the true complexity of life's blueprint begins to unfold. Let's embark on a journey into the cellular hinterlands, exploring how the static code of DNA is brought to life through dynamic processes. This isn't just science fiction speculation, but cutting-edge truth. Our genes are not merely on-off switches, but adjustable dials subject to a myriad of influences. And such discoveries are reshaping our comprehension of health, personality, and disease. Epigenetics doesn't merely add a new layer to our biological narrative, it compels us to rewrite it. We're not predestined by our genes. Instead, our environment, experiences, and choices all play a starring role. From the foods our ancestors ate to the stresses we face today, each factor can nudge our epigenetic dials, sometimes with effects lasting generations. Prepare to unravel mysteries that link aged theories of evolution with modern findings. To comprehend how historic famines speak volumes about our developmental biology, and to recognize the cellular scars left by psychological traumas. If you're ready to venture beyond the simplistic depiction of genetics, to appreciate the shadows and nuances that shape who we are, then let's dive into these nuances. You'll discover that your DNA is merely the beginning of the story. You'll see traces of your grandfather's lifestyle in your genes. Moreover, we'll uncover the poignant lessons hidden in the lives of overtaxed rodents, calling attention to the enduring impact of early life experiences. Welcome to the Epigenetics Revolution, where every gene has a backstory and the past meets the future within our very cells. Part 1. Unlocking Life's Mysteries Beyond the Double Helix. Picture this: humanity finally decodes its genetic blueprint, anticipating a future where every ailment and trait would be traced to a precise spot on our DNA. The completion of the Human Genome Project sparkled with the promise of medical marvels and uncharted knowledge. But just like early mapmakers who could only outline coasts and borders, we realized the map of the genome marked just the beginning of the journey. DNA sequences proved to be the starting point, not the full story. And so we turned the page to a new chapter, the study of epigenetics. Think of it like the difference between reading a recipe and actually baking the cake. The written instructions are crucial, but the quality of the ingredients, the temperature of the oven, and even the humidity in the air all affect the final outcome. This is what epigenetics is about. It's the script notes in the margins of the genetic code, the instructions that tell genes whether to take center stage or wait in the wings. It's both the subtle and the grand adjustments. These factors lead to the variations between genetically identical lab mice and, by extension, in every living organism. Let's delve into their world for a moment. These inbred mice offer an illuminating example as their identical genetic starting points diverge into unique individual characters and physiques. It challenges the simplistic notion that genotype equals phenotype. DNA methylation and histone modification are the backstage technicians of this genetic theater. These adjust the expression of genes, much like a lighting crew influences the mood in a play. DNA methylation can dim the lights on particular genes, while histone modification might adjust the spotlight's intensity, directing our attention to a gene or drawing it away. The early lives of these mice can preset these modifications, with consequences spanning their entire lifetimes. Like green room gossip that influences the performance, what happens backstage in their genes can shape the show they put on. But this isn't just a tale for mice. Our own lives, every quirk, every predisposition, every resilience, are influenced by these same epigenetic switches. The implications for us as humans are vast. 4-minute books could choose to tell the listener that the marks etched by environment, nutrition, and stress can turn certain aspects of our genetic scripts into a drama, a comedy, or maybe even a tragedy. Understanding epigenetics unravels another layer, revealing new paths to explore the vast genetic landscapes within us. It's not just about the genes you inherit, it's about how the story of those genes is told. It's a narrative that unfolds with every breath we take, as much about the actor's marks and stage directions as it is about the original script. Part 2. When starvation writes a script for future generations. Our lives often read like tales scripted by our predecessors, narratives shaped by their experiences, a concept that couldn't be clearer when we explore the aftermath of the Dutch hunger winter. During the final harrowing winter of World War II, the Netherlands faced a horrific famine. The German blockade led to unprecedented starvation, creating a period of such severe deprivation that people resorted to consuming the inedible to survive. Thousands perished, but the survivors, and more importantly their unborn children, became inadvertent participants in an extraordinary episode of human biology. The Dutch hunger winter provided a tragic, large-scale case study on developmental programming, a remarkable process that explores how conditions in the womb can script one's future health. This grim tale of famine revealed stark patterns associated with when, in their pregnancies, the mothers faced starvation. For those expecting, the timing of their hardship inscribed distinct marks on their children's lives. Babies exposed to famine late in pregnancy were often born smaller, a condition that seemed permanent. They generally led lives marked by slightness. However, the normal weight babies whose earliest development occurred during starvation were ironically more prone to obesity in adulthood. These outcomes suggest a profound systemic alteration occurring at the deepest biological level, epigenetics in play. The menacing ghost of famine didn't just threaten life, but configured their bodies to hoard and conserve. Research discovered that the Dutch hunger winter left behind molecular echoes in the form of changed methylation patterns on certain genes within those famine-conceived individuals. While researchers stopped short of claiming definitive causality, a correlation was hard to ignore. Imagine if, during your earliest, most formative stage, your body learned scarcity and hunger as the norm. Your whole system might become incredibly efficient at storing nutrients for the anticipated lean times ahead. Then, when born into a world where food was abundant, that efficiency might translate into an unfortunate tendency to gain weight. The Dutch hunger winter illustrates how our genetic scripts can be edited before we even take our first breath, how prenatal experiences shape the stage for future performances. These methylation patterns serve as annotations from the past, potentially setting the scene for a lifetime struggle with weight, long after the reason for such alterations has ceased to exist. Through this harrowing slice of history, we gain insight into the power of epigenetics. Our genes may be our foundational script, but our earliest experiences, sometimes before we're even born, can annotate that script with profound and lasting influence. Part 3. Do traits acquired in life echo through generations? A historical conundrum revisited journey back 2 centuries, where Lamarckian evolution sparked intense debates in drawing rooms and academies alike. The theory? The traits an organism acquires in life could be bequeathed to its progeny, a concept appealing in its simplicity yet derided by the Darwinian perspective. Picture giraffes, stretching necks reaching for the skies, with each generation standing taller, courtesy of their parents' endeavors. Yet, as Darwin's shadow loomed over biology, Lamarck's ideas receded into scientific folklore, until epigenetics suggested a plot twist in our evolutionary saga. While Darwinian evolution remains our bedrock, epigenetics plays on the boundaries, suggesting that perhaps Lamarck wasn't entirely off course. It turns out, we're capable of passing more than just genetic blueprints to our children. Sometimes, epigenetic scripts, too, can migrate from one generation to the next. Let's revisit the Dutch hunger winter's legacy, not only for the survivors, but for the script it wrote for future acts, the unborn generations. Remarkably, the famine's repercussions echoed into the cradles of the survivors' grandkids. Daughters born amidst that hunger grew up to bear children who weighed heavier at birth, as though the echoes of famine resculpted the gene expression for generations. Now, such observations could be brushed aside as anomalies, mere quirks of the harsh conditions various bodies endured. But when the phenomenon is traced through the lineage of fathers, the narrative pivots sharply towards epigenetics. Unlike mothers, fathers contribute less to the cytoplasmic mix of a zygote, isolated from the pregnancy's dance, making their influence primarily genetic. Yet history whispers another tale of survival and scarcity, this time from the northern reaches of Sweden. Here, bouts of famine were interspersed with seasons of plenty. The effects? Boys who endured hunger before reaching puberty bore children who later defied cardiovascular ailments, while those indulging in abundance witnessed grandsons more prone to diabetes. The plot thickens further when we peer into the rodent world, mirroring human findings. A father's feast or famine impacts his progeny, cementing Lamarckian echoes in the halls of modern biology. The implications extend to our plates, forks poised above a legacy we're shaping with each bite. It's not just a personal affair of diet and consequence, but a familial one, a shared tale from one chapter to the next. Lamarck's centuries-old thesis thus re-emerges, albeit draped in 21st century threads. Our ancestral dining habits, it seems, cast longer shadows than we'd ever imagined. Whether it's a comforting thought or a culinary caution, our daily choices, our acquired traits, may ripple through time, revising gene scripts for generations yet unborn. So as we enjoy the present, let's chew over the notion that we're not only feeding ourselves, but perhaps scripting the scenes for tomorrow's cast as well. Part 4. Exploring the epigenetic shadows of childhood trauma. Tales of struggle and triumph, wherein a child overcomes a traumatic past, often captivate us. They're compelling, heart-wrenching, and reaffirm our belief in the human spirit's resilience. Still, reality casts a darker shadow. Overcoming the long-lasting impact of childhood neglect and abuse is no small feat. The memoirs that end in triumph can misrepresent the truly enduring nature of early hardships, with repercussions stretching far into adulthood. Numerous studies show that individuals scarred by a traumatic childhood are significantly more likely to fall into despair, often succumbing to self-destructive behaviors. The question lingers: what mechanisms inscribe such harrowing experiences deep into the psyche, affecting an individual years, even decades later? The epigenetic hypothesis proposes that the after effects of childhood trauma may be encoded within our very biology, influencing our adult lives from beyond the reach of memory. Among the potential epigenetic culprits, cortisol production stands out, a hormone we associate with stress. Increased levels in those with traumatic childhoods suggest a lasting biochemical legacy of pain and stress. In probing this hypothesis, researchers turn to rodents for clues, gleaning insights from the parenting habits of rats. The diligent grooming and licking of a mother rat to her pups during infancy provides them with solid stress coping mechanisms. Their adult cortisol receptor expression in the hippocampus thrives, acting as a biological totem against stress's corrosive touch. Conversely, the neglected rodents illustrate the darker path. Minimal maternal care correlates to heightened stress in their adult lives, a consequence of reduced cortisol receptors. This epigenetic signature, specifically DNA methylation, essentially silences vital parts of their biological defense against stress. It's as though the playbook for managing life's pressures remains frustratingly out of reach. The parallels to human experience are compelling. If humans follow a similar pattern, if the delicate neuroepigenetic balance can be so readily disrupted, then the narrative of childhood trauma demands a significant plot revision. Our understanding of emotional resilience and fragility might pivot on these epigenetic whispers, passed from one act of life's play to the next. Though research on neuroepigenetics stirs controversy with its radical implications, the potential to uncover the biological roots of psychological pain is too pivotal to dismiss. The hope lingers that, with time and determined inquiry, these enigmatic epigenetic mechanisms might reveal the keys to unlocking a life unhindered by the ghosts of the past, shifting the end of many more real-life stories towards the happier end of the spectrum. Final summary. In delving into the essence of our biological being, our foray into genetics has revealed a complex and intricate story that extends far beyond the helical structure of DNA. While genetics offers a foundation, a script of biological directives, it is epigenetics that brings the drama of existence to life, adding annotations and stage directions to the genetic text. Epigenetics has unveiled itself as a compelling dimension where gene expression is fine-tuned by factors like environmental signals, historical events, and personal experiences. We've uncovered tales of survival, like the Dutch hunger winter, that illustrate how early life malnutrition may weave its influence through generations. We have examined the persistent murmurings of a Lamarckian legacy, where a hint of truth lies in the idea that acquired traits can echo into the genetic chorus of our descendants. Further, we've ventured into the poignant tangle of childhood trauma, exploring how the indelible marks of early experience might be etched into the fabric of our being. The epigenetic narrative suggests that experiences of neglect or adversity leave their signatures on our DNA's performance, influencing our future demeanor and mental health. The revelations of epigenetics have been akin to uncovering hidden notes in a symphony, subtle yet profound. It whispers that our lives are not only the reflections of the genetic scripts delivered at conception, but are continually rewritten, directed, and enacted through the interplays of life itself. With each passing discovery, epigenetics is reshaping our perspectives on health, inheritance, and the very story of human evolution. This field, vibrant and burgeoning, promises to remain under the spotlight, redefining our understanding of what shapes us and how we, in turn, might shape the generations to come. Watch this space closely, for in the theater of biology, the epigenetics revolution may well hold the keys to some of life's most captivating mysteries. Thank you for joining me today on this journey of learning and discovery as we explored the insights of another thought-provoking book in our growing library of knowledge. If you've enjoyed our time together, please take a moment to follow our podcast, give us a 5-star rating, and share 20-minute books with other knowledge seekers. Your support truly means a lot. Don't forget to join me again in the next episode, where we will delve into another enriching book. Until then, happy reading and happy listening.